Hello, good day everyone. This is Group 4 here, and in this lecture, we'll be going through the adaptive immunity, and more specifically, we'll be looking at the cell-mediated immunity. By the end of this presentation, you should be able to define adaptive immunity, explain the role of adaptive immunity, explain what is immunological memory, and give examples. Define and explain cell-mediated immunity. State key cells involved in cell-mediated immunity. State three major ways in which cell-mediated immunity protects the body. Differentiate between humoral and cell-mediated immunity, and give examples of cell-mediated immunity. Have you ever wondered why a child, after being exposed to measles and have successfully recovered, cannot get the virus again? Or why is it that the recovery time for a flu seems to get shorter after being exposed and recovered the first time around? Well, our immune system defends our body against infections. The immune system is broadly divided into two parts, the innate immunity and the adaptive immunity. The innate immune system refers to the body's non-specific line of defense. This system doesn't care what it's killing, its primary function is to kill any intruder. This is the body's first line of defense and includes physical barriers such as skin or mucus. The adaptive immune system, or the acquired immunity, uses specific antigens to bring about an immune response. Unlike the innate immune system that attacks any intruder it sees, the adaptive immune system is activated when exposed to a pathogen and uses an immunological memory to learn and remember the threat so that the next time around, it can get rid of the intruder more efficiently. This system is more specific and efficient as it differentiates between pathogens. However, its response is much slower than the innate immune response, which is always primed to fight pathogens. The major roles of the adaptive immunity include to induce a response this specific to maximally destroy pathogens, to recognize and differentiate between specific non-self antigens in the presence of self while in the process of antigen presentation, to develop immunological memory of an encountered pathogen whereby the pathogen is recalled by a unique antibody that can induce a quick and specific response to eliminate the pathogen should the same infection be re-encountered.
Immunological memory is the ability of the immune system to respond more rapidly and effectively to pathogens that the body has already encountered. The adaptive immune system contains a memory element that enables an efficient response to reinvasion of a particular pathogen. This memory is governed by the adaptive immune response, and there is little dependence on the innate response. When there is an adaptive response to a new pathogen that has never been introduced, this is known as a primary response. It is during this primary response that plasma cells secrete antibodies and differentiated T cells increase. When B and T cells mature, they are now called effector cells. A memory cell is an antigen-specific B or T lymphocyte that does not differentiate into effector cells during the primary immune response. However, they can immediately become effector cells if there is re-exposure to the same pathogen. During the primary immune response, memory cells do not respond to antigens and do not contribute to host defenses. When the infection is cleared, the effector cells are no longer useful and they die. Memory cells are formed when B cells find an antigen that matches its receptor, then waits for it to be activated by a helper T cell. Then the B cell divides and produces a plasma cell and a memory cell. Plasma cell produces antibodies that attaches to the current types of invaders. Eater cells such as macrophages prefer intruders with antibodies and eats all of them. If the same intruder invades again, memory cells can help the immune system to activate faster. B and T cells will go around the body for a few years, and if the pathogen does not come into contact during the person's life again, they will gradually die off. If the host is exposed again to the same type of pathogen, the memory cell in circulation will differentiate into plasma cells immediately. The adaptive immune response is delayed because it takes time for the B and T cells with the correct antigen to be identified and activated. If reinfection occurs, this step is skipped and therefore this results in faster production of immune defenses. Memory B cells that differentiate into plasma cells produce vast amount of antibodies than that secreted during the primary response. The fast and dramatic antibody response can stop the infection before it can even become established. An example of immunological memory of vaccinations. A vaccination for a virus can be made using either an active but weakened virus or specific parts of that virus that is not active. The weakened virus and the virus particles cannot cause an infection. Instead, they mimic the presence of an active virus so that an immune response occurs, even though there are no real threats. So, by getting vaccinations, what you are doing is exposing your body to the antigen that is required to produce the antibodies specific to that virus and acquire a memory of the virus without experiencing any illnesses. Reinfections create more memory cells, so this increases resistance to the pathogens. In some cases, memory cells can die and an additional booster shot may be needed to mimic exposures. An example of this is tetanus, which is recommended every 10 years for adults.
What is cell-mediated immunity? Cell-mediated immunity is an immune response that does not involve antibodies. Instead, phagocytes, antigen-specific cytotoxic T lymphocytes. What are the key cells involved in cell-mediated immunity? They are T and B lymphocytes. However, T lymphocytes can be subdivided into helper T lymphocytes, cytotoxic T lymphocytes, and suppressor T lymphocytes. These two types of lymphocytes look similar but have developed in different ways and thus have different functions. They are both formed from stem cells in the bone marrow. However, B lymphocytes develop in the bone marrow and T lymphocytes develop in the thymus gland. Each of the lymphocytes has their own unique protein receptor sites found on the surface of the membrane whose molecular shape matches one specific antigen. However, T lymphocytes constitutes about 80% of lymphocytes in the blood. They do not produce antibodies. Instead, they respond in many complex ways. One particular way involves producing T killer cells, which directly destroys infected body cells. This process is known as a cell-mediated response. T helper cells help other parts of the immune system to kill the infected cells. Memory cells retain a memory of the specific antigen, which provides immunity. Both B and T lymphocytes do not work in isolation. They work together in the removal of pathogens. Cell-mediated immunity has T cells, which recognize infected cells within the body and eliminate them before the intruder can replicate and infect other cells. T cells cannot recognize these pathogens without help. An antigen-presented cell, such as a dendritic cell or macrophage, detects, engulfs, then digests pathogens into antigen fragments. The fragments are now transported to the surface of the antigen-presenting cell, where they are presented on proteins referred to as major histocompatibility complexes class II, often referred to as MHC2. T cells will be activated towards an antigen once they encounter the antigen displayed on an MHC2. When a virus or bacteria enters the cell, T cells are activated when encountering an antigen on the infectious cell that is presented on the MHC2 of the antigen presenting cell.
And humoral immunity, as briefly stated by the picture below, we have the substrate molecules, which are the antigens. The AB cells are triggered when it's encountered itself with a matching antigen. They are then engulfed into this molecule. The B cell engulfed the antigen and digested it. Then we have the combination of antigen and MHC attract the help of mature matching T cells, right? As you can see by the cell-mediated immunity complex there below. Afterwards, cytokinins are released into the bloodstream and antigens and antibodies are secreted, right, by the liver and spleen in all bodies of animals and etc.
Cell-mediated immunity examples: graft rejection. Grafts are living tissue that is surgically transplanted from one part of the individual to another, or from one individual to another, for its adhesion and growth. Grafts, unless it's donated by an identical twin, is usually recognized as antigenic by the receiver's immune system. The receiver's immune system responds to this and results in the destruction of the graft. CD4+ as well as CD8+ T cells are involved in graft rejection and respond to differences occurring between both the donor as well as the receiver of their class II and class I histocompatibility molecules. Graft versus host disease. In order to provide or restore a source of blood cells for a recipient, grafts of bone marrow are used. Many cancers are treated vigorously through cytotoxic chemicals, as well as radiation, which results in the patient's bone marrow being destroyed. This is where grafts of bone marrow come into play to help restore the patient. It can help in treatments to remove cancer cells, or it can be used to restore the patient's bone marrow. If the marrow is taken from a donor that isn't an identical twin, or the receiver themselves, there is no danger of rejecting the graft, as the recipient has no functioning immune system. There will be, however, histocompatibility differences between the receiver and the donor, where the T cells from the donor will mount an immune response against tissues of the recipient. In this case, immunosuppressive drugs are used to control graft versus host diseases. Contact sensitivity. There are many people who experience rashes on their skin when in contact with certain chemicals. Some examples include certain dyes, nickel, and the active component of poison ivy plants. This response is triggered by the CD4+ T cells and can take 24 hours to occur. The reaction between the chemical to the proteins found in the skin is what creates the antigen. Dendritic cells work in the skin to engulf the antigen, then they migrate to neighboring lymph nodes where fragments of the antigen are presented to the CD4+ T cells. T cells are now activated and migrate to the skin to induce an inflammatory response. To test your knowledge on adaptive immunity, the cell-mediated response, here is a short multiple choice quiz. You can pause the video, and when you think you have it, resume the video for the solutions. Here is a quick summary of the content discussed in today's presentation.